like to thank you for your attention, and I would be happy to answer any questions.
Dear Mos, thank you for your presentation. Now we have questions from our participants. The first question is, they said, my question is about the replication factories. Are they similar to the factories induced by different positive standard RNA viruses?
Well, they're not similar in with regard to the proteins, but the basic strategy of focusing all the viral proteins involved in replication in one area is efficient, so that many viruses will have a similar strategy.
Thank you. The next one is, what do you suggest us as undergraduate students who are willing to study in virology in the future?
Oh, that's very difficult to predict the future. I think it's important to have experience in two areas. Bioinformatics is very important now because we're able to accumulate so much data that from sequencing and for large-scale expression studies, that you have to be familiar with computer analysis. So that's one part. But I would hope that the future generations of virologists will not do all their experiments on the computer, but will still be able to go into the laboratory and look at the real virus, not a computer virus.
Thank you. The next one is, can you address clear regulatory requirements for sequencing?
Requirements for what?
Requirements for sequencing.
Quarantine, yeah. Okay, of course, we know now, we didn't know in the beginning that individuals who didn't have symptoms would be able to spread the virus. But now we know that the majority of infected individuals either don't have any symptoms or the symptoms are just like a common respiratory infection. So therefore, quarantine is necessary to see whether somebody who has been in contact actually has the infection. Now, with more testing, it would be possible to shorten the period of quarantine. If you don't do any tests, then people have to be quarantined for 10 to 14 days. If you had more testing, you might be able to shorten the quarantine just to several days.
Okay, thank you again. Now, the last question is, why can we get immunity from smallpox with one vaccine in our life, but we should take the influenza vaccine every year?
Yeah, well, that is a question that immunologists are trying to understand. And that was one of the reasons we thought vaccinia virus might be a good carrier for vaccines for other pathogens. What we really don't understand is, although immunity can last for 50 years after vaccinia virus, there's no evidence that the virus remains in the body. So there are memory cells in the immune system which propagate for long periods of time, and they replenish themselves, so they're able to continue to have immunity. Now, why some viruses make more of these memory cells and other viruses make less is not clear. But there's another factor with influenza virus, and that is because it changes. So therefore, the old vaccine no longer works. Now, with smallpox,
according to Wikipedia, DNA sequencing is the process of determining the nucleic acid sequence, the order of nucleotides in DNA.
Years, at least not to a significant extent. So you didn't have to change the vaccine. And it also gave this long-lasting immunity. But there's hope that with further research, we'll be able to prolong the immune status after any vaccine.
I said last question, but we have one more question. So it's, what are the possible dangers against monkeypox pandemic?
What is the danger against a poxvirus pandemic? Is that what you said?
What are the possible dangers against monkeypox pandemic? It says monkeypox.
Oh, monkeypox pandemic. Okay. Yeah. So right now, there's not a serious problem because, as I said, it doesn't spread from human to human. If it mutated like coronaviruses to infect better, then of course we would have to start vaccinating everybody against it. And the smallpox vaccine is already being used in Africa to protect people against monkeypox. So if it would start to spread, we would already have a vaccine.
Dear Dr. Barnett Mose, thank you for your informative presentation and answers. And also thank you for participating in our event. It's an honor for us to listen to you.
Well, it was a pleasure for me and an opportunity to speak to young students in Istanbul. Thank you.
Again, thank you. Dear our teachers and guests, the 18th International Molecular Biology and Genetics Winter School has ended with Dr. Barnett Mose's presentation. We hope we will be together again next year. Thank you so much for your participating. Stay with science.